In this middle third of our course, we've been exploring topics related to the dual conceptual themes of development and homeostasis. But as I've admitted to you several times now, we've taken a fairly animal-biased approach to this material. That is, we've explored questions about development, homeostasis, internal communication, and physiology in the context of animal systems, largely ignoring other major groups of multicellular organisms, most notably plants. Now, there are a couple of reasons why we've been focusing on animals in this middle part of the course. The first reason, perhaps a selfish one, is that we ourselves are animals, and naturally we're interested in how we work when we're asking questions about form and function. A second reason is that some aspects of form and function in organisms are simply more elaborated in animals. This is especially the case, for example, when we think about internal communication mechanisms such as the nervous system, mechanisms involved in sensory transduction, or mechanisms involved in the ability to move. Now, a third reason is that major groups of organisms, plants versus animals, for example, do differ in some very fundamental ways that make it difficult to generalize across groups, at least on some levels. We were able to consider plants and animals in similar terms, for example, when we talked about molecular genetics or Mendelian trait transmission or evolutionary processes and so on, at the very beginning of the course. For example, how DNA is replicated, transcribed, and translated into proteins works pretty much the same way in plants as it does in animals, at least to a first approximation. Similarly, we used Mendel's pea plants, you'll remember, to serve as a primary example for understanding trait transmission from parents to offspring in sexual reproduction. On the other hand, plants and animals obviously differ in fundamental ways when it comes to their structure, how this structure develops, and many of the physiological mechanisms that are used to maintain this structure over an individual's lifetime. But I certainly don't want to leave you with the impression that we can simply ignore these kinds of processes in plants, even if they differ from what we've seen in animals. Thus, in this lecture and the next, we're going to take a tour of form and function in plants. In a sense, what we're going to do is go back and cover some of the same conceptual ground we've just gone over in this middle section of the course, but we're going to look at this material from a different perspective, from the perspective of how plants work instead of how animals work. Now, before we start exploring how plants function, we have to begin by considering something about their structure. Plants are multicellular organisms, of course. As I mentioned earlier in the course, the evolution of multicellularity occurred independently in plants and animals over 1 billion years ago from different unicellular ancestors. So given this distant relation, it's not surprising that the structure of plants differs radically from the structure of animals. Actually, what might be more surprising to us as we go along is how much plants and animals have in common. Let's start our discussion today by considering how a single plant cell differs from an animal cell. Now, plants, like animals, are eukaryotic organisms, and so their cells actually have many more similarities with animal cells than they have differences. Like any typical eukaryotic cell, plant cells have a nucleus, they have an endomembrane system, and they have a variety of other membrane-bound organelles with specialized function. By and large, there are really only 3 major differences between plant and animal cells that we have to consider, but these are major differences. Now, the first and the most significant difference between plant and animal cells from our point of view is that in addition to being surrounded by a lipid bilayer membrane, plant cells also are enclosed in a rigid cell wall. This cell wall is composed of cellulose, which is a very stable sugar polymer, a polysaccharide, which is embedded in a matrix of other kinds of polysaccharides and proteins. Actually, the structure of a plant cell wall is a little bit like reinforced concrete, with the cellulose forming very rigid reinforcing rods within a matrix. Now, the plant cell wall actually provides an even greater degree of protection for the cell than a cell membrane alone does, of course. It also provides structural rigidity to the cell. The most obvious consequence of plant cells having a cell wall is that plants don't need skeletons to hold themselves up. Instead, the ability of plants to stand upright against the force of gravity largely has to do with the structural support provided by the cell walls of the plant's own cells. In the case of very large plants, there are actually some kinds of cells that develop especially strong walls and enable the whole plant to achieve enormous heights in spite of an equally enormous weight. All you have to do is consider, for example, a mature redwood tree or a giant sequoia to imagine what can be achieved this way. Now, another consequence of the fact that cells have walls is that individual plant cells actually can't move relative to each other in a tissue. And this relative inability to move, as we'll see, has important consequences for the way plants develop and actually the way that they otherwise respond to the environment. Now, the two other main differences between plant and animal cells that we should note are that plant cells have two kinds of organelles that animal cells don't have. One of these kinds of organelles I've already mentioned to you when we first introduced eukaryotic cells, and this organelle is called the chloroplast. Chloroplasts are one of the two organelles we mentioned as energy processing organelles, the other being mitochondria. Chloroplasts are the site, as we'll learn later in the course, where the process of photosynthesis occurs. Photosynthesis is the way that plant cells actually take energy from sunlight and use that to manufacture sugars, which they can store for energy, use as food, or use to build other kinds of organic molecules. Both plant and animal cells have mitochondria, as we'll see later. Those are the organelles that actually break down organic material to make energy, but only plant cells and also the cells of some photosynthetic unicellular organisms have chloroplasts. Now, the second kind of organelle that's unique to plant cells is called the central vacuole. The central vacuole of the plant is basically one large chamber on the inside of the plant. It's made up of elements of the endomembrane system, so it's a big membrane-bound open space. It's largely used for storage. A plant cell might store materials for nutrition in there. It might store, for example, pigments in there if it's a colorful cell, for example, a cell in the petal of a flower, or it might store waste products in there. The central vacuole also plays a very important role in the way that plant cells grow and change their shape, because this central vacuole can very rapidly absorb water and in so doing expanding, and by expanding it can press out against the cell wall. And as we'll see, if the cell wall then becomes less rigid in one dimension or another, the cell will expand in that dimension. We'll see how that plays a very important role in development and actually in the way that plants can essentially move in response to outside stimuli. Okay, now that we've seen how plant cells are distinct, let's consider the structural organization of a typical plant. That is, let's look at how plant bodies are put together. Now, plants as a group are extremely diverse as animals in many dimensions, so of course it would be impossible to describe a common structure of plants that really applies very cleanly in all cases. But if we limit our discussion to a group of plants known as the angiosperms, these are what we would commonly call the flowering plants, then we actually can come close to sort of a simple common description of a body plan. Angiosperms, as I said, are flowering plants, and this group of plants is actually the most diverse and widespread groups of plants that live on land. It's a very recent evolutionary divergence. So focusing our discussion on of plant structure on angiosperms is a reasonable focus for our discussion of plants in general. Now, the fundamental structure of land plants, such as angiosperms, reflects their need to exist at the interface of two very different environments. One environment is the ground. The ground is where plants are going to anchor themselves and also where they're going to draw water and other resources, especially minerals. The other environment, of course, is the air. Now, the air is important to plants because that's where they get carbon dioxide, which, as we'll learn later, is an essential part of the photosynthetic process. Basically, carbon dioxide is where the carbon comes from that the plants will use to make more complex sugars. The air is also where essentially plants can be exposed to light. Again, the source of energy that plants use for photosynthesis. Because they live at this interface between the ground and the air, actually the fundamental organization of plant bodies is divided into two main components. There's what's called the shoot system, which is the part of the plant that's above ground, and the root system, which is the part that's below ground. Now, roots and shoots are specialized in very different ways in terms of their overall morphology and specializations of the cells that occur within them. The point that we want to keep in mind right now is that as the plant develops, what is going to become part of the root system has to differentiate in very different ways from what's going to become part of the shoot system. The shoot system actually involves generally more complex structures as well as compared to the root system. For example, parts of the shoot system will turn into complex organs such as leaves, which are specialized for exposing surface area to gather light for photosynthesis, or the reproductive organs of plants called flowers. Now, for each part of a typical flowering plant, whether it's shoot or root, we can define three major types of tissues. We refer to these types of tissues as dermal tissue, vascular tissue, and ground tissue. Dermal tissue, which is also sometimes called epidermis, is usually represented by just a single cell layer on the outside surface of a plant. Dermal tissue is essentially like the skin of an animal. The cells of a plant's epidermis are especially durable. They're specialized to prevent injury or invasion by pathogens. In some parts of the cell, actually, dermal cells also may be specialized to take in or release materials, especially parts of the epidermis associated with the root system are specialized to absorb water and minerals from the soil. Now, vascular tissue runs essentially along the center of the plant, and it includes two types of tissues that are specialized for transporting materials and conducting them from one part of the plant to the other. One of these tissues is called xylem, and it's specialized to transport water along the length of the plant, primarily from the roots up through the rest of the shoot system. And the other vascular tissue is called phloem. Phloem is actually responsible for transporting sugars and other materials manufactured by the plant, largely from the leaves down to storage areas in the rest of the plant or into the roots. In this sense, vascular tissue, as its name suggests, is sort of a plumbing system for the plant, roughly analogous to the function of the circulatory system in animals. Now, ground tissue makes up the rest of the plant. Ground tissue is a much more diverse kind of tissue functionally. You can think of ground tissue as essentially being a tube of tissue running along the length of the plant. If you picture a plant body as an elongated tube going from the root up to the shoot with branching tubes off as those roots and shoots branch off, you can imagine if you cut through the center of this tube, what you would see is on the outside, the epidermal tissue. On the inside, in a more complex structure, you would see vascular tissue, and in between those two, you would see ground tissue. Ground tissue performs a variety of functions. It especially serves to provide structural support, so cells of the plant that are going to be specialized to provide more structural support with having thicker or stronger cell walls will occur largely in ground tissue. And it's also a place where cells are specialized to store food or other compounds manufactured by the plant. Now, one last point I want to make about the structure of a typical plant is that as compared to animal bodies, at least to a first approximation, there appear to be fewer specialized organs with highly complex structures. If you take apart a vertebrate animal like ourselves, it's just filled with all sorts of convoluted, specialized, different parts and tissues combined in what we call organs. If you take apart a plant, you generally see less than that. Nonetheless, plant bodies do have specializations among their parts, most notably things like leaves and flowers and other kinds of specialized organs. This point then brings us back, really, to a question we asked much earlier in this middle segment of the course. How does a complex multicellular organism, in this case a plant, develop from just a single cell? How do we get all of the different cell types in the right combinations and in the right arrangements occurring in the development of a mature plant? And this is the question we're going to focus on for the remainder of this lecture. Now, our overview of the organization of a typical generic flowering plant body suggests a few specific questions that we should ask about how plants develop. First, I've already mentioned one, and that is to ask, how is the root to shoot axis established? What is it that determines this asymmetry between structures that are going to become parts of roots and structures that are going to become parts of shoots? Actually, this shoot to root axis, if we turn that around, is more technically referred to as the apical basal axis of the plant. Apical being the top of the shoot part of the plant and basal being the bottom of the root part of the plant. Now, a second question we might ask is, how are these three basic tissue types that characterize the parts of the plant established? Again, this question is analogous to one we asked when we considered development in animals. You remember that we saw that early on in animal development, three fundamental types of so-called embryonic tissues were determined. These are the ectoderm, the endoderm, and the mesoderm. How do these kinds of tissue types arise in plants? Now, after we consider these questions, what we're going to do is turn to a third, more specific question concerning the development of specialized organs in plants. And here what we're going to do is look at genetic mechanisms that underlie the development of the specialized morphology of flowers. So let's begin. Just as we saw when we discussed animal development, the development of a complex multicellular plant from a single cell, which is the fertilized zygote, depends on really three major kinds of processes. It has to depend on cell division, making more cells, cell differentiation, those daughter cells turning into different kinds of cells, and morphogenesis, cells occurring in the right numbers in the right places in the right parts of the plant. By and large, the role of these three processes in plant development is similar to their role in animal development, but with two major differences. The first difference has to do with differentiation. Fully developed adult plants, or fully mature plants, actually have significant amounts of tissue that are made of relatively undifferentiated cells, which retain the ability to develop into many different parts of the plant. This is unlike animals, you'll remember, where undifferentiated cells are relatively rare. In animals, we call these cells stem cells, and those stem cells occur in particular kinds of tissue where we need to generate new cells throughout our lifetimes. But those stem cells are also relatively determined to only be able to develop into a few kinds of cells that are important for the tissue in which they exist. By contrast, plants retain a large number of cells that have much more potency to turn into different kinds of things. These undifferentiated tissues in plants are called meristem tissues. Meristem tissues occur especially at the tips of growing shoots or growing roots. We call these apical or basal meristem tissues. And meristem tissues also occur in a cylindrical band that runs along the tube that would be our generalized plant body, which allows for growth, lateral growth, essentially expanding that tube. Now, the second major difference in the developmental mechanisms used by plants and animals has to do with morphogenesis, and this isn't surprising. The cell walls of plants render them incapable of moving around relative to each other. Remember that when we looked at animal development, we saw that the migration of cells plays a very important role in morphogenesis. For example, we saw how migration of cells actually established the three fundamental tissue types during the process of gastrulation, where cells from the outside of a hollow ball moved to the inside and changed the shape of that hollow ball, essentially establishing the three major tissues and the head-to-tail axis of the animal. This kind of cell movement doesn't occur in plant development, meaning that the role of morphogenesis in plant development must be limited largely to patterns of cell growth. Now, with this kind of general background, let's take a quick look at how the apical-basal axis of a plant is established in development. The very first division of a plant zygote, right, a fertilized male and a fertilized female gamete fertilized by a male gamete, the very first division is very asymmetric, producing one small daughter cell, which will eventually turn into the embryo, that is, turn into the plant, and a much larger cell, which actually will form a nutritional support that's used to support the developing embryo. This nutritional support is something we call the suspensor. Now, this first division actually also sets up the basic shoot-root axis by creating an asymmetry that's outside of the growing embryo. As that smaller daughter cell begins to itself divide and form a ball of cells, those cells that are adjacent to the suspensor organ that's developing will be determined to become root structures, whereas cells on the other side of the growing ball will become determined to become shoot structures. Now, the specific molecular mechanisms responsible for this process of determination in plants are still poorly understood, but it seems clear that cell-cell interactions between the embryo and the suspensor are responsible for this early specification of the apical basal axis. For example, in one model organism, which is a small weed used a lot in plant development studies called Arabidopsis, this is a member of the mustard family, there's a mutation of a gene called NOME actually leads to a symmetric first cell division. And if this first cell division is symmetric, the suspensor fails to develop. And if the suspensor fails to develop, the growing embryo never establishes a shoot-root axis, even though other aspects of development proceed relatively normally. Now, the three basic tissue types found in plants also were established very early on in development, just as they are in animals. Again, we see here a rough analogy to animal development, in which we have a ball of cells that eventually becomes an outer layer of cells becoming dermal cells, a middle layer of cells that in this case will become ground cells, and a central part of the growing embryo that will be determined to become vascular cells. But this analogy is only a rough one. First of all, the ball of cells of a growing plant is not hollow, it's solid. And as I said, there are no movements. This is called the globular stage, descriptively in plants. There are no movements of cells that help set up these different tissue types. Instead, again, it seems as though there must be some sort of positional information, cell signals that tell individual cells where they are relative to the center or the outside of this ball of cells, and in so doing determines their fate to become one of these three different tissue types. Again, the specific molecular mechanisms are still the subject of much active research. Now, if a plant cells don't move in development, we have a general question of how any kind of morphogenesis can lead to more complex structures. We won't go into this in any detail other than to point out that the way morphogenesis occurs in plants largely has to do with changes in cell shape. Now, we saw that changes in cell shape were also important in animal development, as animal tissues folded and evaginated to create different parts of complex organisms, or to create limb buds as the animal was developing. Morphogenesis in plants is entirely dependent on changes in cell shape, and very briefly, the way these changes occur are by signals that cause different parts of the cell wall to become less rigid or more flexible. These cell signals basically change the rigidity of the cell wall and then trigger the cell to expand by an increase in the size of the central vacuole. So, for example, if the cell wall becomes less rigid in one dimension and not the other, then as that central vacuole expands, it will push the cell out. And you can imagine we can have a cell that elongates in one dimension or another dimension, depending on the orientation of how the cell wall loses its rigidity. These kinds of changes in cell shape are the mechanism by which all of the organs that have complex shapes in cells, by and large, arise. But I don't have time to talk about the details of that. Instead, I want to turn to a more specific question, which is how flowers develop in plants. Now, to give you a little background about this, I need to tell you what flowers are. Actually, flowers are a collection of 4 different organ types, arranged in 4 concentric circles, or what are called by botanists whorls. Now, the outermost whorl of a flower is made of structures that we call sepals. Sepals are sort of the green leaf-like things that enclose a flower bud before it opens, and as the flower bud opens, these sepals will bend back, revealing the rest of the flower. The next ring in includes what we call the flower's petals. Petals, of course, are the colorful or showy parts of most flowers, and they're often specialized to attract insects to come in and pollinate the flower. If we continue to the next ring on the inside, we have organs that are called stamens. Stamens are the male reproductive organs of a flower. They're the organs that produce male gametes, which are called pollen. And finally, the innermost ring or whorl of organs in the flower are called the carpels. These are also referred to sometimes as pistils. Carpels or pistils are the female reproductive organs. This is where the female gametes are produced, and actually, as it turns out in flowering plants, this is where fertilization occurs and the zygote begins its development to create a seed. Now, the question we want to ask is simply this: how does an undifferentiated piece of meristem, for example, an apical meristem, a meristem that's on the outer edge of a growing shoot structure, become determined not just to produce a flower, but to produce these 4 distinct flower parts in the right arrangement? The first insight into how this happened actually came from studying mutant strains of plants that had malformed flowers. For example, in the Arabidopsis plant I mentioned a moment ago, there are 3 such classes of mutants, with each of these classes being characterized by lacking 2 out of the 4 types of organs that make up a normal flower. One class of mutants, for example, has only stamens and carpels, lacking sepals and petals. A second class of mutant has only sepals and carpels, lacking petals and stamens. And a third class has only sepals and petals, lacking the stamens and the carpels. Now, the details there aren't important. What is important is the fact that there are 3 classes of mutations suggested early on that each mutation is caused by a defect in a single different gene. It further suggests that the 3 genes responsible for these mutations, when they're defective, must code for transcription factors that initiate a cascade of other regulatory genes, which are those the more complicated set of genes, the cascade that's necessary for the development of each organ type. Thus, these genes, which are involved in flower organ development, are roughly analogous to the kind of genetic master switches that we saw when we looked at segmentation genes and homeotic genes, when we looked at Drosophila development. But the question is, how can 3 regulatory genes be responsible for controlling the development of the 4 different organ types in flowers? Well, in brief, the answer to this question is embodied in what's become known as the ABC model of flower development. The name of this hypothesis derives from the fact that the 3 genes involved were initially named just simply gene A, gene B, and gene C. There are 3 basic tenets of this model. First, the model assumes that each of these 3 genes is expressed in 2 adjacent rings or whorls in the meristem that will give rise to a flower. Remember, we have 4 rings that we have to account for. Second, the assumption is that this pattern of gene expression somehow will cause these 4 rings to turn into 4 different kinds of organs. Third, and finally, the model assumes that each of these 4 combinations of these master regulatory genes are going to, as I said, trigger a cascade of other gene expression, which is what leads to the specific flower part. Now, the way to visualize this model is simply as follows. Imagine that you're looking down on a cross-section through an apical meristem that's going to develop into a flower. You have 4 rings to keep track of. Gene A, the model goes, is expressed in the outermost 2 rings. Gene B is expressed in the middle 2 rings. So in the second ring in, we have both B and A expressed, for example. And gene C is expressed in the center 2 rings. So we have in the outermost ring only gene A expressed, and presumably if only gene A is expressed, that triggers the development of sepals, the outermost organ. In the next ring in, we have gene A and B expressed, which triggers the development of petals. And then if we go 1 more ring in, we have B and C expressed together, which triggers the development of stamens. And in the innermost ring, we have only C, which triggers the development of carpels or pistils. Now, there's one more assumption that this model makes, and that is that gene A's product inhibits the production of gene C's product and vice versa. And with that assumption in mind, let's go back and consider what we would expect for mutations of these genes. If gene A is lost because it's mutated, then the gene product for gene C is now expected to be produced in all 4 of these whorls or rings, because gene A is no longer inhibiting it in the outermost rings, and therefore we have gene C through all rings. What that means is that because gene C is produced in the outermost ring and in the innermost ring, and because gene C is responsible for the production of carpels, or also known as pistils, then we would have these carpels on the outermost ring and the innermost ring, and now in the 2 middle rings we would only have the production of stamens, because we now only have the expression of gene B and gene C. Similarly, we could go through different combinations of mutations and predict what kinds of organs you would expect to see in these mutant plants. And the interesting fact is that when you actually look at these mutations, the combinations of organs and their position in the mutant plant are exactly what are predicted by this distribution of gene products. Well, this overview of how plants develop actually leaves us with an interesting perspective on the interplay between the unity and diversity of all living things. Plants are organized very differently as compared to animals, and it's not surprising that they're different in many aspects of their structure and function. At the same time, many of the fundamental principles by which they work, especially with regard to the role of cell interactions in development, positional signals in development, and the function of master regulatory genes, largely are all the same, at least in general principle, even if the exact players are different. This leaves us with the suggestion that there's a really fundamental commonality in the basic mechanisms that can be used to build any kind of multicellular organism.